Hello,
Hello.
how are you?
I am well. Good to meet you, even if it's virtual.
That's right. Good to meet you too.
Thank you.
S-
Thanks so much for the invitation.
Oh, not a problem. Yeah, great to actually be able to hear about your work.
Thank you very much.
So let's have you get, yeah, so you should now be able to.
At the Georgia Institute of Technology in 2013. He was chair of the former department from 2014 through 2024. Professor Finn's current interests include the use of virus particles as molecular and catalytic building blocks for vaccine and functional materials development, the development of quick reactions for organic and materials synthesis, polyvalent interactions, and advanced linker technologies in drug targeting, and the use of evolution for the discovery of chemical function. He is currently the Chief Scientific Officer of the Georgia Tech Pediatric Innovation Network, which brings new science and engineering to the aid of pediatric medicine. Graduates from the Finn Laboratory can be found in academic departments around the world, as well as in companies in the pharmaceutical, biotechnology, chemical, and materials industry. Professor Finn was the first recipient of the annual Scripps Outstanding Mentor Award, a 2017 Arthur C. Cope Scholar Award, and was editor-in-chief of the journal ACS Combinatorial Science from 2010 to 2020. So help me in welcoming Professor M.G. Finn.
Thanks so much. Thanks to Professor Murray and the other organizers. I'm really delighted to be with you and honored for the invitation. So thanks. Want to, so I can't see a chat or anything. So let me just invite anyone if you have a question or a comment, just go ahead and interrupt. It's totally fine.
Oh, I set up
Fine
the
with me
chat
anyway.
now. I have the chat open now.
Okay, great. And I'll encourage Dr. Murray to just break in if there's a question that you want to ask. And I'm going to tell you not quite a fully current story. I'm going to do a little bit of history for you to kind of try to introduce you to the idea of what click chemistry is, and then give you some examples of how we apply both the technology and the philosophy in doing science and engineering with molecules. So, of course, my favorite thing, of course, is working with wonderful students. I have just an amazing group, and here's a very recent picture. Those who are working most intensively on some of the themes that I'll talk about today are the ones highlighted. Graduate students all, with the exception of Jasmine, who's a postdoc. Wenting, Val, Jeff, and Devashish are all working on various areas of applications of click chemistry to immunology and material science. Here's an older slide, just so that I didn't want to leave out two former members of the group, Robert and Parisa, who are very important to some of the stuff I will tell you today. And my dog Scout, who is always with me, and she's right here in the office, if you want to say hi to her. And lastly, a couple of our undergraduates. So this is our current undergraduates in the lab, but Kalia and Ava are also working on some of the stuff that you'll hear a little tiny bit about toward the end. So I'm very proud to represent this wonderful group. So we're going to talk a little bit about click chemistry, which was really part of the Nobel Prize of 2022, awarded to Barry Sharpless and Morten Meldal for the development of a particular reaction, Sharpless for also the development of the idea of click chemistry, and Carolyn Bertozzi, the wonderful Carolyn Bertozzi, for development of the corresponding or adjacent idea of bioorthogonal chemistry. And I really obviously won't have a chance to talk about the contributions of all of these wonderful people, but I do want to talk about click chemistry and a little tiny bit of its history. So in the 80s and 90s, which is sort of the gestational period for this idea, drug discovery was not really going very well. This is a plot of the number of new drugs approved as a function of years. You see it's steady between sort of 20 and 30, the uptick here is significant. That's actually biologic agents like the antibody therapies that you see on TV all the time. But most of these new drugs weren't really new. They were knockoffs or slight variations of previously approved drugs. The drug discovery industry was really not really coming up with a lot. And so, what do you do about this? And academic and industrial chemists started to adopt a new strategy, a different strategy, which was instead of trying to design every candidate drug that you wanted to test, the idea was let's just make lots of molecules and make them quickly and then figure out how to test them so as to try to find agents that might do what you want to do. So this picture of a young John Ellman, this picture was taken when he was an undergraduate working with me as a graduate student. John went on to currently, of course, a very distinguished career at Yale. He was one of the first to think about and to describe this idea of making lots of molecules via combinatorial chemistry. Combinatorial biology had already started to take off, and that really is the science of making large libraries of molecular agents, such as by phage display, the development of George Smith. Francis Arnold adopted methods to do combinatorial development of enzymes, that is, catalysts for chemical reactions. And Greg Winter here adopted the idea of phage display and enabled it to be used to make humanized human antibodies, as you see in this title of this picture. So if you happen to be unfortunate enough to go on one of these antibody therapies that you see advertised all the time, you can thank Greg Winter for that, among many others. But the whole philosophy here is to let the biological engine make lots and lots of molecules and try to pick the ones that do what you want them to do. So combinatorial chemistry, as started by Ellman and many others, and you may already know this, is the art of making lots of different candidate molecules with a minimum number of chemical reaction steps. So here's a pictorial illustration of that. If I start out with a collection of 3 starting materials, and I do 3 reactions, the same reaction, but each with a different building block, I will then come up with 9. If I pool all those 9 molecules and then do 3 more molecules, subjecting the pool to a second reaction with each of 3 different building blocks, now I have 27 distinct different molecules, and so on. If I do another step, now I have 81. So I've done just 3 chemical reactions, but I have lots and lots of different molecules that presumably will have different properties. And so as you start to get into this whole field becomes now quite exciting and lots of people doing work in it. And there was an appreciation early on, however, that the number of molecules that could be made was just staggeringly large. This is a lovely treatise of that. It's a computational exercise. The estimate was that if you restrict yourself to common elements and a reasonable small molecule molecular weight, the number of molecules that might exist that could be made are in the order of 10 to the 63rd. The number of molecules at the time that had been synthesized and characterized was only about 20 million. And so this number is just staggering, right? If all 8 billion people made 1 trillion compounds a day, it would take 10 to the 32 billion years to make all of these molecules, right? So the space that we're trying to deal with, and this is again in the context of small molecule drug discovery, the space is unimaginably large, right? And in fact, nature does not do it this way. So if you think about it, nature is by far the best inventor of new molecular properties that we know of, right? Biology makes amazing molecules and families of molecules that do everything that we know about, right? That do the most difficult things. And nature does not play a numbers game, right? Here's a slide from one of my introductory lectures for biochemistry, right? This is how nature makes functional molecules, polymers like proteins, right? Puts together monomers into a larger structure by doing a very reliable reaction to connect building blocks to make functional molecules, right? Nature does this, of course, with polynucleotides. Same thing, same philosophy. Monomers linked together with reliable chemistry. And indeed, nature does this for polysaccharides. All of its most complexly functional molecules are made this way. And of course, nature, to adapt those molecules to change their properties, nature doesn't make a whole new family. Nature does evolution, right? Programming the content of the molecules into a system that can be tweaked by cycles of evolutionary change. So just making a lot of compounds might not be the way to go. And so Sharpless asked, really was the real creator of this challenge. What could we do instead of trying to make lots of different molecules? What could we do if we restricted ourselves to the molecules that are easy to make? And if you have a reaction that makes molecules easily, then in fact, you can use that reaction principle to make a diverse set of molecules using that chemistry. That was Barry's challenge. And our inspiration for this, I was involved in the early stages of kind of thinking about this idea, which was just delightful. One of our inspirations was the chemistry of polymers, right? Modern life cannot happen. Our society would fall apart if all the oil went away, right? In addition, of course, to poisoning the planet. A real crime about burning up all the oil is that oil, petroleum, has all of the building blocks that we need for modern science, and for modern life, right? So one takes, for example, a small molecule like propylene, polymerize it, and you make a lot of different kinds of materials. Here's acrylates, polyacrylates. Again, you make large molecules from small building blocks. Those are all sorts of other kinds of materials. Here's ring opening polymerizations like polycaprolactone. Those are used in a lot of medical devices. Everything we use in modern society comes at base, or many of the things comes from polymers, which derive from crude oil. So there's lots of different function here. There's not a lot of different building blocks, right? So this is the inspiration for Sharpless's coining of the idea and the language of click chemistry. The challenge was, if you can find reactions, chemical reactions, that make bonds very reliably, no matter what's connected to the parts of the molecule that's doing the bond forming. If you can find those reactions and use them relentlessly, can you make functional molecules? Because if you can, and if they're made only using reactions that are highly reliable, then you have no worries about scaling things up, or about making variations, or doing all of the difficult things that we chemists in the medicinal chemistry game and the material science game that we worry about. Click chemistry, reliable reactions, provides you with a lot of power if you can find and use them. So in the early days, we always thought of click chemistry as spring-loaded. And the reason was that the quintessential reactions were things like epoxide ring openings, right? Lots of energy built into the epoxide unit. But if it's a terminal epoxide or a terminal aziridine, right, if X is nitrogen, or a terminal episulfide, if X is sulfur, really these molecules will only do one thing. They'll only react with nucleophiles and open at the less hindered carbon. They won't do elimination, they won't do condensation, they'll just do ring opening. Spring-loaded, lots of energy built in, but they only do one thing, which means you can drive a lot of reactions that you might have trouble otherwise doing with a lot of different kinds of nucleophiles in this case. Lots of other reactions turned out to meet those criteria. The criteria were reliable bond formation, typically carbon heteroatom bond forming, not carbon-carbon bond forming. Right? Nature makes carbon-carbon bonds reliably, really wonderfully, but with a level of control that, frankly, we chemists still cannot match. Right? The chemistry of the aldol condensation reaction, one of organic chemistry's favorite carbon-carbon bond-forming reactions, is very complex. Right? Aldol reactions can go in a dozen different directions in the laboratory. Nature controls aldol condensations with enzymes that took, 1 billion years to evolve, right? We don't have that kind of control typically for the kind of applicability that we're talking about here. So typically, click chemistry makes carbon heteroatom bonds, often high energy, pre-programmed. By that, I mean a single kind of reaction. building blocks. When you find these kinds of properties, the reactions have really very no workup or very innocuous byproducts. And interestingly, and it's the subject of a whole nother lecture, many click reactions actually work best in water. So the challenge was, is function achievable using these kinds of reactions as much as possible? Let me pause. Did I hear something? Anybody have a question? No? Okay. All right, so here was the paper we wrote now long ago. I'm sure well before many of you were born. It's in the audience. But the idea was, again, to kind of lay out this philosophy and also show some examples. So a process chemistry point of view, process chemistry is what chemical industry does when they know what they want to make, but now they need to figure out how to make a lot of it inexpensively. Process chemists are the champions at finding efficient reactions. Right, olefin-based organic synthesis, because a lot of the building blocks came from carbon-carbon double bonds, lots of reactions in water, etc. So this idea, I'm going to skip this. So this idea then got crystallized into a collection of reactions that were already very well known, and we wanted to call attention to them as paragons of the kinds of power that one can have in chemical bond formation. So again, strained rings, right? These three-membered rings. Some interesting electrophiles like aldehydes react very reliably with certain nucleophiles, but not all nucleophiles, right? Sulfonyl halides, an interesting class. Alpha beta unsaturated carbonyls do Michael reaction, conjugate addition chemistry very reliably, right? Diazonium chemistry, still beautifully and underdeveloped, I think, diazonium reactions. And then there's a collection of reactions that involve cycloaddition processes, right? So like the Diels-Alder reaction, typically not very fast, but very selective, right? It does only one thing. And the champion of click reactions or click chemistry very quickly became the azide functional group. The azide group is really unique in science, unique in earthbound chemistry, in that it is a 1,3 dipole. Right? If you imagine the positive charge here and permanent negative charges on the N nitrogen. So it's a dipole, maybe not a 1,3 dipole, but a permanent dipole. And it's stable, right? You can put it in a bottle, you can keep it on the shelf. There aren't many reactants that react by dipolar cycloaddition that have those properties. And so the 2022 Nobel Prize could have been called the Nobel Prize of the azide functional group. And so as we were thinking about click chemistry, of course, most of the very good reactions had already been discovered, right? Old chemistry is very often great chemistry. And much of it was buried in the old German literature, you know, from the 40s and 50s and even before then. This is a paper from 1893 by Arthur Michael, the same Michael of the Michael reaction, describing actually the cycloaddition of alkynes, written weirdly, right? We would write this as a carbon-carbon triple bond, right? But you see 2 electrons here, 2 electrons here, and 2 electrons there. It is a carbon-carbon triple bond. And the azide, which is, again, they didn't really understand exactly what the structure was, right? Azides look like this, but that's okay. Azide plus alkyne gives you this 5-membered ring, a triazole, and that was, again, first discovered or described in 1893. The person who really understood and popularized this chemistry was another German, wonderful Rolf Huisken, who I have had the great privilege to talk with. And he really popularized the azide-alkyne reaction. And it still wasn't really widely used because it gave 2 different isomers, right? If you see the R group here is next to the phenyl ring and here on the opposite side, and it was pretty slow. You had to heat it up. So click chemistry really took off when these 2 papers were published within a month or 2 of each other. And this is what won Martin Meldahl and Barry Sharpless the Nobel Prize in 2022 for the discovery of copper catalysis of this reaction between an alkyne and an azide. Right. So these groups worked independently. They didn't know what the other was doing. They both reported this fairly close to each other. And it turned out to be really a game changer. And so my group has spent a lot of time working on this reaction, just trying to discover how it works, which I'll tell you a little bit about, But the reason why it has been so useful, and if you ask somebody what is click chemistry, very often they will point you to this reaction. But there are other click reactions that I just showed you. But the reason why this has really become the quintessential click reaction is because of how useful it is. And it's useful because of this thing, this bullet point that I have here first. Azides and alkynes are very small, just 3 atoms and 2 atoms, respectively, forgetting about that terminal hydrogen there on the alkyne, right? And these functional groups are very small, and they're not polar. They're not acids. They're not bases. They don't do pretty much any other kind of chemistry other than this cycloaddition reaction. So they are therefore invisible. They're not highly polar. They're not highly nonpolar. And so when you attach them to something, they typically do not change the properties of the thing that you're attaching them to. So you then have what we'll call an invisible piece of Velcro, right, that you can attach to something. And when you want to make a bond, then you have to give it the right catalyst or the right reaction conditions. They're easy to install. Organic chemists are very good at putting azides and alkynes onto things. This reaction is actually downhill by about 50 kilocalories per mole, but it's very, very slow. So I should pause here and ask your organic chemists, those of you taking organic chemistry, how that can be true. And of course, the answer is that thermodynamics does not necessarily imply kinetics. There's a large energy barrier to go over, right? The activation energy of this reaction is large, even though the thermodynamic driving force is large also. That is a very unusual combination. And it gives us then an opportunity to control this reaction by changing the nature of the reactants. In this case, if you make the alkyne electron withdrawing or strained, then the reaction speed will pick up, or by catalysis, or by environmental constraints. And by that, I mean holding them together for long periods of time. You can get the reaction to happen. So I'm going to tell you just a little bit about both of those kinds of things, and then I'll rocket through some applications so you can see why it's useful. So at the time that click chemistry, that this reaction was discovered, my group was trying to actually do organic chemistry with viruses. Nobody had ever done that before. And I have models of viruses all over my office here. I can pick one up and show you. This is a 3-dimensional model of a virus, kind of one of the ones that you see here. And what this is a 30 nanometer particle, huge from a chemist's point of view, right? 2 million molecular weight. But we can make a lot of it for reasons that I will not describe now. And what we were trying to do is actually make bonds at particular spots on this kind of highly symmetric structure. So here you see lysine residues on the surface of the proteins, where they are and how far apart they are and all of that. So we took advantage of the special reactivity of cysteine, sulfur, a unique nucleophile in biology. And we by mutation, we put sulfurs in, put cysteine residues in particular places. And you can see their different patterns. And we did reactions with malleimides, with electrophiles that were supposed to react with cysteine selectively. And we got some lovely structures. What this is a virus structure. This is a plant virus engineered to have cysteines at 60 positions. And what we did was we reacted it with a gold cluster that had a malleimide group on one part of the gold cluster. And we got a beautiful arrangement of gold in a icosahedral form on the surface of this protein assembly. We published a lovely paper. It caught a lot of attention. It was really fun. And it turned out it had a chemistry lesson in there that was very important to us later. So I'm zooming in on that structure I showed you. We're zooming in on one of these gold clusters. By the way, the green here is RNA that's on the inside of this virus particle. Kind of cool. And so this is this gold cluster, and the gold cluster should be spherical. It's actually not oval shaped as it appears here. And we were wondering why that was, why we got an oval shape. And what we thought was this structure was determined by X-ray crystallography. And so we thought there was a little tether and the gold was just vibrating, was just rocking back and forth. And in X-ray crystal structures, that will often average out to something that looks like, something larger. But it turned out not to be the case. What was really going on was we had a reaction at the cysteine, and then in 25% of the cases, a reaction at a neighboring lysine, that is an amino group, was reacting about 25% as fast as a thiol group with this reagent that should only have reacted with thiols, according to the books, according to the literature. So what that told us is even this very good chemistry wasn't good enough to be absolutely specific. So I'm actually going to skip this. I'm already going over time here. So we immediately applied this azide-alkyne reaction to doing reactions on the surfaces of virus particles, and we got perfect attachments, 60 in this particular case, 60 attachments per particle, with a very high yield of the overall structure. And that means that we were doing chemistry very efficiently, right? 99+% reactivity yield at each of those sites was what was necessary to give us this kind of yield. And there was no reaction that we had known of previously that could do this on a biological molecule. So that was the breakthrough. And so we were able to incorporate natural amino acids into these proteins very easily using methodologies developed by colleague Dave Terrell at Caltech. And away we went. So we've studied this reaction. We figured out how it works. And I'm actually going to skip a lot of that because, again, I'm running over time. But the power of this chemistry needs to be emphasized a little bit. This is a scheme here that illustrates a complicated situation. We have a virus. We're attaching to it hundreds of different alkyne groups, in this case, not using an unnatural amino acid chemistry, but just regular old amine reactivity with an activated ester, right? Amines on the surface. But now this puts alkynes all over the surface of the virus. We need to now attach a number of different molecules. In this particular case, we're trying to study how a particle covered with different sugars interacts with cells. So we now have the power, because we have a highly reliable reaction, we have the power to take molecules that are really hard to make, like this tetramannose derivative or this octamannose derivative, or even this nonamannose derivative. These are all synthetic molecules made with an azide group, and we can attach them in different numbers to this protein scaffold by using this reliable copper chemistry. And so it's completely predictable. It doesn't matter how big the molecule is that's attached to the azide, they all react the same, because the fundamental reactivity of this azide with this little 2-carbon linker here, or this 4 or 5-carbon linker here, that chemistry is entirely reliable, predictable, the rate constant we know, and we can just mix these molecules and get exactly what we want, 470 plus or minus 50 of all of these molecules here, or 230 on the structure, just because we just mix them in the right ratio. So it really is, and again, no other chemistry can do this, or at least could do it at the time. So the reaction is a beautiful, it's a wonderful organometallic reaction. I won't have time to step through this, and I'm just going to skip all this, I'm sorry. But for those of you interested in mechanistic organic chemistry or mechanistic organometallic chemistry, I'm happy to talk about this if you want to contact me offline, because it's a beautiful story, a very interesting story. But just to suffice it to say, I'm just going to skip to the end here of this little vignette. This is kind of captures what we understand about this reaction. This particular reaction, and it was not designed this way, requires 2 copper ions to be organized in space. And this is the kind of ligand, this central nitrogen with 3 copper binding arms on it. If you can see that molecule, that's the catalyst or the pre-catalyst, but you mix it with copper, it spontaneously forms this kind of structure. And you need 2 coppers because one of them binds the alkyne by making a copper-carbon bond, and the other one has to lean over and tug on the pi electrons of the alkyne to activate it to react with the azide, which is bound to this first copper ion. Right, so azide binds, this formation of the copper acetylide here is really fast. Azide binds reversibly, makes that linkage. The system is pre-organized then to make this 5-membered ring. And then this catalytic cycle is completed by replacing the copper-carbon bond with a carbon-hydrogen bond, typically from water that's around in the environment, right? So this reaction chugs along, but it turns out if you have something else that binds copper competitively, this whole catalyst can disassemble into some copper solvated species. The ligand again has to reassemble and come back into the catalytic cycle. So it turns out the kinetics of this reaction are really complicated, but it's a pretty simple concept, pretty simple reaction when you get right down to it. Okay, so I'm skipping some of this stuff. Here's now another click reaction that is widely used, and again, discovered many years ago. This was 1961. This is another variation of an alkyne cycloaddition, but in this case, it's a strained alkyne doing now a very fast Diels-Alder reaction on what is a very interesting diene, right? But strained alkynes then turned out to be interesting. And I want to teach you one more concept about reaction selectivity that turned out to be very useful. So here's an example of a need that the drug discovery industry would have. I want to create a molecule that binds selectively to a particular protein, let's say an active site of an enzyme, and I don't exactly know the structure of the protein, right? So I can't design a molecule to fit the key. I can't I can't design a key to fit the lock, I should say. So what do? Well, one strategy is to give the protein lots of different building blocks and see which building blocks bind, and then try to create molecules that join those building blocks. But if you have a chemical reaction, X and Y, or reactants that can actually join themselves, not too fast. You want them to join only when they're held together on the surface that you want to develop a binding agent to. So if you can do that, then you can incubate a combinatorial library of building blocks and let the target self-assemble the molecule that binds best to the target. I hope that makes sense, because the azide-alkyne reaction is just perfect for this if you have some patience. Remember, I said that the azide-alkyne reaction is 50 kcals downhill, but very slow. It's slow enough that if you took what is it? I think it's a 10 millimolar solution of azide and alkyne, or maybe a 100 millimolar solution of azide and alkyne, unactivated, and just let them sit in solution, it would take them 40 years to do the reaction, if not catalyzed. But if you hold them together in close proximity, right, that means the concentration, the effective concentration, is very, very high, then the reaction can go more quickly. So we demonstrated this years ago in the early days of click chemistry using a protein target called acetylcholinesterase. When you get a snake bite, if you're unfortunate, the snake bite toxin, the snake venom toxin, is an inhibitor of this enzyme, right? It's actually a femtomolar inhibitor of the enzyme. And acetylcholinesterase helps your neurons function by hydrolyzing acetylcholine. So it's a highly active enzyme. And so we made a femtomolar inhibitor, and actually even better inhibitors, just by this strategy. And it took quite a while to figure it out, because the molecules were so tight binders, they couldn't come off. And so we actually couldn't do the analytical chemistry. But what we did was he incubated azides and alkynes, and all we looked for was the formation of a molecule that joined the two, because we knew that they would never form unless they were templated on the target, right? And so the target itself then gave us the answer, and we hadn't pre-designed the answer. That kind of selectivity is quite rare, and is now being used quite a lot in the chemical industry, and we called it in situ click chemistry. And here's a couple of examples of subsequent papers that use that to make lots of different things. Okay, so again, I know I'm skipping because I want to leave time for questions. So we now have this azide-alkyne reaction as an example of a very powerful molecular joining function. What can we do with it? So we do lots and lots of things with it. Here's an example from Material Science. So this is a paper we published a couple of years ago, where we made building blocks, you see here, with two different alkynes on them. And if you think about the chemical structure of this building block, you see there's that carbon atom in the middle, which is a quaternary carbon, right? So this is what's called a spirocyclic compound. And if you can imagine, I should have had a model built for you. I'm sorry. I don't think I have it here. Probably not. But if you think about the three-dimensional structure, that spirocyclic molecule, right, is like this, if you can just look at my hands, and it can't be flat. Right? There's no way I can flatten this molecule, right? So it's three-dimensional. And we use these kinds of building blocks to make polymers, right? Long connections of molecules that cannot pack on themselves. And so they are intrinsically porous. They have porosity built in. So in this case, we took different building blocks with two different alkynes. We married them with different building blocks with two different azides. You see the azide groups, right? And we make polymers just by mixing them with copper catalyst. And what that makes are polymers that actually we make membranes out of. You see here, this is a polymeric membrane on a porous support. Here's the kinetics of the polymer reaction. The polymer gets made very quickly. The polymer membranes then actually are able to molecularly separate the components of crude oil. You heard me talk about crude oil by virtue of their size, and are they aromatic or not aromatic? Why is this important? Well, as I mentioned to you, crude oil has thousands of compounds in it. Many of them are highly useful. We make medicines out of them. We make polymers. We make all sorts of things out of them. And of course, we also make gasoline, right? We shouldn't be burning it. But 1%, a full 1% of the energy used by human species on Earth is the distillation of crude oil. 10% of all the energy used by industry on Earth is the distillation of crude oil. We use a phenomenal amount of energy to get the energy out of this material. If you could instead filter the components instead of heating them up to distill them, you can save 90% of that energy that you're using to distill crude oil. So this is why we do this, why we develop these materials, is to try to get rid of a lot of the distillation by separating out, let's say, the small stuff from the big stuff, and you can then use a lot of that material directly. So here's an example of stuff that comes through. This is a two-dimensional GC analysis. In this case, we worked with the evil people at ExxonMobil. They are not evil. Their company, we don't like them to burn so much fossil fuels, but they're really wonderful scientists there, and they helped us out a lot with this. So we can take a crude oil that looks like this, run it through the filters, and get very nicely low molecular weight components that are then very useful in a number of applications without having to distill. Here's another example of how we use click chemistry. Very different, right? Here we're talking about carbohydrates, that is sugars, and we're very interested in developing vaccines against things that make you sick, like bacteria or cancer. And as it turns out, every cell in nature is covered with carbohydrates, and many of the cells that make you sick have unique carbohydrates. But the problem is carbohydrates are usually not seen by the immune system. Why? Because they all look pretty similar, right? If you know what a carbohydrate is, it's a carbocyclic ring with a lot of hydroxyl sticking out. Now, synthetically or spatially, three-dimensionally, carbohydrates are the most diverse structural class in biology. They're amazingly structurally complex. But they all look kind of the same too, right? They're just polyhydroxylated material. It's also not a good idea to make an immune response against your food, right? So we are evolved not to have an immune response against most carbohydrates. However, if I could teach the immune system to respond to a particular carbohydrate that's on, let's say, a bacterial cell and not on a mammalian cell, then I have a very interesting way to make a vaccine against that bacteria. Hope that makes sense to you. So we have developed a way to do that. And the way we do that is to synthesize the carbohydrates. Oh, look, what an unexpected pleasure, with an azide on them, right? And we attach those carbohydrates to virus particles of the kinds I showed you before. And when we do that correctly, and we use the right virus particle, it turns out that viruses are very immunogenic. In other words, our immune systems are evolved to respond to viruses, and we can hijack that. We can take advantage of that, put disease-related carbohydrates on a platform like this, and have the immune system say, oh, I think this is a molecule I want to respond to. And so we can make vaccines, and we, in this case, make high-affinity antibodies, or the mice do when we give these constructs to mice, make high-affinity antibodies to something that previously they did not recognize at all. And so we make vaccines or vaccine candidates from this kind of thing. Here's another application example, and I'm almost done, where we wanted to make antibodies to different variations of the drug fentanyl. As I'm sure you all know, fentanyl is incredibly dangerous, right? Far more potent than heroin, and kills, tens of thousands of people a year in the United States alone. And the reason is, unfortunately, it's too easy to make. From an organic chemistry point of view, this is fentanyl. I'm not going to show you how to make it. But if you're an organic chemist, you can probably imagine how it's made. And there are hundreds of variations of fentanyls out there. And unfortunately, the test kits that hospitals have and that law enforcement has does not detect a lot of those fentanyl variants. And this is a paper we published with the CDC showing that many of the dangerous fentanyl derivatives are not detected by the test kits that a police officer might have if they find a white powder somewhere and they need to know what it is. And so we embarked on a project to make antibodies that recognize a lot of the different variants of fentanyl. And we did that by synthesizing different components, the things that look like fentanyl with different structural attributes. Oh, look, we put an alkyne on each one of them. We put those alkynes on virus particles. We sent those virus particles into mice, and we got antibodies to each of those structures, different structures. This shows the antibody response. Again, I know I'm skipping through this very quickly, but the antibody responses are incredibly good. And then we made antibodies that detects, now blue is good here, and here are 30 of the most dangerous fentanyl variants, that detect all of those fentanyl variants with at least one of our antibodies. And so in principle now, we can detect now far more of those compounds that are dangerous to people out in the field. Okay, I think I'm done. I'm going to skip this whole other kind of chemistry here, and I'm going to just go to the end, to the lessons. So remember I told you that 10 to the 63rd thing, right? There are an unimaginably large number of molecules. Well, if you think about a molecular problem, let's say how I can make an antibody to fentanyl, right? Or how can I stop an enzyme that is a key enzyme in the processes of a bacterium so that I have an antibiotic agent, right? Those are all molecular problems. Nature will solve those molecular problems by evolution, as I told you. But if you think about it, the structure of a molecule, the functional structure of a molecule should be I should be able to mimic that with a lot of different kinds of molecules, right? If I need to put a carboxylic acid here, an amine group here, and a hydrophobic group there are probably millions of answers to those problems, right? To each problem, many molecular solutions are likely to exist, right? If you use, in the search for those solutions, truly reliable reactions, the more you use reliable reactions, the more you use click reactions, as we've called them, you have fewer reactions to choose from, right? However, you have many more building blocks you can use because the reaction should be much less sensitive to what is being carried on the backside of the functional groups that are being joined in making that bond, right? Azides and alkynes will join no matter what they're attached to, for example. So what that gives you then, if function derives from connecting those blocks, like it does in proteins and polysaccharides and polynucleotides, the biological inspiration, or polymers, right? If function derives from connecting blocks, then click chemistry allows you to make many diverse blocks, right? So click chemistry and polymer science are intimately related. Click chemistry and biological polymers are intimately related in concept. And in principle, we in the laboratory, by using these reactions, have access to just the same kind of power that nature does. Diversity, as it turns out, is much more important than numbers when you're talking about making molecules, or I would argue, talking about any organization involving different individuals. You want to bring in components that have many different kinds of properties, right? So diversity is key, and click chemistry allows you to access the maximum molecular diversity that you can think of. When you use click chemistry, now synthetic chemistry becomes a little easier, actually a lot easier. So lots of people can do it. And then testing becomes more important than making things. Discovery shifts from being synthesis limited to design and screening limited. You still got to be a good chemist to figure out how to use these reactions. You got to be a really good chemist to discover new reactions like this. But then you also need to talk about or think about how you design the molecule construction and how you test for its function. Molecular solutions made with click chemistry are instantly scalable. I can make lots of them because the reactions work well. Discovery of new click reactions, I didn't talk about that, but that's really now the beautiful part, right? The hard part. If you can discover a reaction that has these qualities, you've really done something. And now a whole new world opens to you. Okay. And once you have them, as I illustrated far too quickly, and I apologize for that, you can go in many different directions. My laboratory does immunology and material science because we have good chemistry that we can use. So I'll leave it there, open the floor for questions, and thank you again very much for inviting me.
Well, thank you. Thank you very much.
Thank you so much for your presentation. I'm now opening the floor to any questions that anyone might have about his presentation today.
Yeah. Like when you're picking getting an immune response for some of those alkynes, you explain that segment. I don't understand how you're able to take an alkyne, attach it to a virus molecule, and then get the immune response to then recognize the alkyne by itself without the virus molecule.
Got it. Right. So no. So okay, yeah, no, great question. The alkyne is used to make the connection, right? And then what we have is the molecule we want the immune system to see arranged on a virus particle, right? And the virus particle has many copies. So here's my virus particle, many copies of that molecule all over the surface. So your immune system is actually evolved to recognize structures like viruses that are what we call multivalent, that have lots of copies of the same thing on them. Why is that? Because viruses often have that kind of structure, because viruses are often made by small molecules that self-assemble multiple copies to package the genome that's inside. And so this polyvalency is a natural feature of things that make us sick. And so our immune system recognizes these particles by actually a number of different molecular sensors that recognize multiplicity in structure. The viruses are, because they're the right size, again, we're evolved to take particles that are of this size, and they're trafficked very efficiently to the lymph nodes, which are in your body, multiple places. The lymphatics drain and take viruses to lymph nodes, and lymph nodes are where the immune system does much of its work. Okay, what happens is a virus particle is engulfed by what's called an antigen presenting cell. That antigen presenting cell then rips open the particle, the virus particle, takes little chunks of the protein and presents them on what's called MHC, major histocompatibility complex proteins, on the outer surface of antigen presenting cells. And so those little fragments of viruses are displayed, and the immune system says, okay, is this something I recognize or something I don't recognize? The act of ripping it up and displaying it, when we attach our desired antigens to the viruses, they just get carried along for the ride. And if the chemistry is very reliable, so that we place that molecule all over the virus particle, now biology looks at what we have attached as essentially being a natural component of the virus. And so we're tricking the immune system to recognize something artificial by attaching it to a surface that the immune system sees as a platform for potential hazard. And then we get to hitchhike all the rest of the immune system's mechanisms by using the reliable chemistry to attach the antigen to the particle. Does that make sense?
Yeah.
Okay. All right.
Hope so.
Go ahead. Follow up.
So then is there a way you could short circuit the whole system by then just simply skipping to the steps of the chunks of a virus particle? Can you then take a virus particle, split it apart outside of, and then study what that is, find out what that is, and then replicate that structure and send that instead?
Okay, another good question. There are two reasons why that's usually not a good idea. Okay. One is by putting it on the virus particle, I didn't explain all sorts of other things that happen that are advantageous because the antigen, the target, is arrayed on the virus particle. And what those are different mechanisms by which the immune system activates itself to say, hey, there's a problem. So if I were to pre-disassemble the particle, I would lose the cell signaling and cell communication events that happen because I'm presenting the antigen on a particle. And some of that actually is physical connectivity. So as I told you, the particle gets eaten, but also some of the particles will bind on the outside surface of a cell, let's say a B cell, and another cell will come along and recognize and bind. And the mere fact that those two cells are connected, physically connected by a polyvalent structure that bears the same antigen, there will start to be some communication that happens that will tell the T cell, hey, turn into a kind of cell that helps me out here, or proliferate to create a killing phenotype or a signaling phenotype. So one of the many ways in which a particle helps you is by actually being big and polyvalent, because again, that's a feature that we're evolved to recognize. There are other aspects of the immune response that also benefit from that kind of structure. So the short circuiting that you're talking about, excuse me, is actually a really good idea. And actually, we're trying to do this now. But what we're trying to do is not replace the particle, but introduce an additional component that basically feeds the immune system the little chunks that we want in addition to the particle, okay, at the right time. So I like the way you're thinking. Oh, and by the way, let me quickly, just 2 sentences. This is the great thing about crossing fields. Okay, I'm a chemist, right? I'm not an immunologist. We do a lot of immunology now, but I ask questions just like you did. You're the student who just spoke up. That's a chemistry question, right? Why not chop it up into a little thing? It's more efficient. A biologist would never think that way, right? But chemists think that way, and we can bring that to a new field and maybe make an impact. So good on you.
So I have a couple of questions. Could you give us a short version of compare and contrast between click chemistry and the bioorthogonal chemistry? How are they
Sure.
similar and how are they different?
So they're very, very similar. Bioorthogonal chemistry is largely a subset of click chemistry, by being applicable and applied to biological molecules and biological systems. Okay, bioorthogonal chemistry tends to be as reliable, or maybe the better word is as selective, as click reactions are, because that's the essence of bioorthogonality. Orthogonal means I'm orthogonal. I don't react with anything else in biology. I only do what I want to do, right? And that is, in essence, a part and parcel of click chemistry. Now, Caroline Bertozzi's development or initiation of bioorthogonal chemistry was actually, those reactions were inspirations for click chemistry and vice versa. So all of that was happening at the same time.
So I also noticed that the catalyst, the copper catalyst, is copper 1.
Mm-hmm.
It looks like it started as copper 2 sulfate, and then the ascorbate reduced it.
Correct.
So copper 1 is absolutely necessary. Copper 2 cannot do that.
Correct. Yes, that's exactly right. Copper 2 and copper 1 might as well be different metals entirely.
Wow.
Right? They are completely different in their properties.
Okay.
Copper 2 plus alkynes will couple alkynes together in what's called the Glaser coupling reaction, and actually make copper 1. So that's one way you can actually generate copper 1 from copper 2.
Okay. And also, have you found that sulfur, I'm pretty sure you guys, did this work. Does sulfur inhibit the copper?
That's an excellent insight. Yes, it does. And so that is one of the major impediments to using the copper catalyzed version of click chemistry in vivo.
Okay.
Right? So in fact, you cannot do, well, this is a topic of really hot area now, or has been for a while, people trying to do this. But in general, copper is toxic when unregulated inside cells because it generates reactive oxygen species. And so I love copper. Copper is one of the most fascinating stories of biological control you will ever see. There's tons of copper in cells, 50 micromolar, 100 micromolar, but there is no free copper. It is completely regulated down to the attomolar level, right? Often by thiols, glutathione, but much more by a set of copper binding proteins that compete with glutathione and pass copper around. Imagine this, it's a system that has attomolar binding constants, but fast exchange. Totally unique in biology. It's really just a wonderful story. But anyway, copper one is the engine, and free thiols do dramatically inhibit the reaction.
So are there efforts now to use different catalysts, and how is that working out? What are some others?
Yeah, so the azide-alkyne, metal catalyzed azide-alkyne chemistry is still at the speeds you need it to be done, is still restricted to copper one. Ruthenium complexes will do it, but much, much more slowly. And so the copper chemistry has really been all about trying to make ligands that protect the copper from inhibitors like thiols. But this field has moved on to other reactions that don't need copper. And the disadvantage is the fragments that you're using are much larger, so they're more perturbing. But many people find that fine. So that's the chemistry of what's called copper-free or strain-promoted click chemistry or azide-alkyne chemistry. And that's what many people, including us, use in vivo.
Okay. Any other questions? You had one sort of like I would describe as a throwaway sentence and moved on, which was that you said that diazonium chemistry underdeveloped. I caught on that. I'm assuming that you have some ideas about how to develop it some more. Are you guys doing stuff like that in your lab too?
Yeah, so no, my lab is not doing that. I've seen some stuff from friends and collaborators that's coming out. I'll just tell you, diazonium reactions are so beautiful because, of course, super high energy, right? But stable in water, right? And people just think of diazoniums as unstable. They are not unstable, right? You can put them in water, they'll live for a good long time, and the reactivity, when you give it the right nucleophile, is off the charts fast, right? So people just haven't thought of it except in the context of azo dyes, right? You make a diazonium salt, right? And it turns out even that reaction is actually wonderfully useful. That is, well-known chemistry, Matt Francis and others in biology, well-known bioconjugation chemistry. But there's a lot of other chemistry of diazonium salts, I think, that remains to be developed.
Sounds good. All right. Any other
question?
Okay.
I was just curious how wide you were talking about, like the cancer and the fentanyl. Like how widely used are those ways to, like, the one that the cancer one where it's like to trick the immune system into detecting it, and then the detection of fentanyl, like how widely are those used?
So we are trying to advance a vaccine on the principle I told you against a parasitic diseases, a particular parasitic disease called leishmaniasis. The philosophy there is the same. The target is a sugar, and we have, I think, really lovely data that shows that we have a vaccine candidate. So, it's going to take us a few years probably to get that into human clinical trials. But that's the farthest developed from out of our laboratory for vaccine development using the methods I talked about. The antibodies are sort of easier because it's just a molecule, right? An antibody is a molecule, not a vaccine. And so the CDC has actually used our antibodies, which we developed along with them. We're in the same town. So our antibodies were used in the first 2 years of the COVID pandemic for the CDC as the analytical standards. So when they were when they were assessing antibody response to COVID early in the pandemic, then our antibodies were the kind of the standards to which those were compared. And so we're very proud about that. They're not commercially available, but the sequences are published, and people can make them if they need them. We haven't wanted to make any money off of that. So stuff is, it takes a while to get to the marketplace, and most of the time it doesn't make it to the marketplace, but hopefully it helps other people do what they want to do.
So that's the work that you do with the leishmaniasis. That's a tropical
Yes.
disease, and hopefully we'll be able to get you back to talk about that. That should be very interesting work.
Very happy to talk about that indeed. It's fascinating chemistry and biology.
Right. And it will have a great impact in, tropical areas around the world.
Yeah.
Yeah. Fascinating stuff that you're doing. You probably can't wait to get up in the morning to go to lab.
It's a privilege to have this profession, and the profession is under attack right now, but we'll get through it.
We hear you. All right. You want to close it off?
Okay, so let's thank him again for joining us this evening.
Thank you all very much.
Right, we'll stay in touch.
I hope so. Thank you.
All right.
And if anybody has any questions, just send me an email.
Yeah,
Sounds great.
some of our students got inspired to maybe do graduate work with you. I don't know. We'll see.
Yeah.
Yeah.
There's lots of good stuff to choose from, my lab and lots of other labs. So I know you prepare them well. So thank you for doing that.
All right. Very good. Very good. Okay, then. Take care.
Thank you. You too.
All right. Bye-bye.
Bye now.